Thank you.
Hello and welcome. My name is Mitch Sanders. I'm the Communications Chair for the Wound Healing Society, and we're delighted to have another Fireside Chat session. And today we have two very special guests. We have Proma Basu, who's a postdoc in Manuela Martin Green's lab, and Manuela's here as well. And we're talking about probably one of the coolest papers we've seen in the Wound Healing Society, Wound Repair and Regeneration sometime, using systems biology approaches to identify signaling pathways activated during chronic wound initiation. And welcome to you both.
Thank you.
Thank you, Mitch, for the nice introduction. I appreciate it. And yes, let's go for it.
Let's begin. So before we get started, I always love the human side of things. So Manuela, if you could talk a little bit about your background, your journey as a wound care expert, and how your path to being, studying wound care over the years.
Well, so I was born in Africa, in Angola, and had to run away with my family to Portugal during the Civil War. There I studied through college. Then I got a Fulbright to come here to the States and initiate my PhD. And in the middle of the whole thing, I met my husband. So I got married and I stayed. My PhD is actually in developmental biology, but then I went to a postdoc in Midabissil's lab as a cell biologist. And then I went as an adjunct assistant professor to Rockefeller into the lab of Ida Saburo Anafusa for the microbiology and for the molecular biology. After that, I got a job at UCR as an assistant professor. And interestingly enough, you asked me, how did my career start in wound healing? It started back in Bissel's lab when I was working on trying to understand how Rust sarcoma virus causes tumors. And in the process, I discovered that this gene that had actually been isolated in the laboratory of Ida Saburo Anafusa in Rockefeller was actually chicken IL-8. And I showed in many different ways that this molecule, which we just called chicken IL-8, was a very important in wound healing processes. And then later on, when we got the sequence of all the chemokines, we realized this is really a chemokine, like we know today. IL-8 is not the real interleukin, it's a chemokine. Then that's how I started. I started studying basic processes of wound healing and the function of chemokines. And then as the time I became associate professor, so I was a little bit more safe in my career, I decided, okay, I'm going to switch from chicken to mice, and I began to study processes of healing in mice with the idea that somehow, somewhere, sometime, I would develop a model of chronic wounds. And so that's what I did. And actually, I went on a sabbatical leave to NCI, and then another sabbatical to Jeff Gertner's in Stanford. And that's really what gave me the time to think about the process of healing and what might be that could cause the wounds to go chronic. When I came back, I began to put all of that in action, and was fortunate enough to have a good graduate student who helped me implement my ideas, and in fact decided and we were successful in obtaining a model for chronic wounds generated in mice, in diabetic mice, and that was published in 2014. Of course, since then we have used this model in many different ways to be able to understand how chronicity occurs. The one that we're going to discuss today with the help of Proma, she's a bioinformatician, and I hired her because of that, is one that I think is going to give us insight. And of course, this is just the beginning. We are doing much more work to try to understand how wounds become chronic. So that's in a nutshell. Coming, and I've stayed with UC, which is, of course, the loyalty tax. But I like the UC, and I'm now at the top of my career as professor. So
that's exciting. It's funny how we all have our journeys. I started in cell biology as well a long time ago. Did my PhD with Yuli Wang, and then did 2 postdocs at MIT with Paul Metzeler and Julie Theriault. And I studied cell motility and cell skeletal interactions and host pathogen interactions with Listeria. And then after I left my postdoc, I started my first company, and I was going to make a Band-Aid, actually a frowning face on your hot dog package if your food was spoiled. And I realized nobody in the food industry wanted you to know you had salmonella or listeria on your chicken. And I had to pivot and said, well, I'll make a Band-Aid change color if you have an infection. And that's how I got involved in wound care to start. So why don't we get going with this paper? It's just amazing. Can we first start with the model, the diabetic mouse model that you developed in your lab in 2014, and the antioxidant enzyme inhibitors to make the wound more chronic, wounds more chronic?
So let me. So we know that when injury occurs, we have a raise in oxidative stress, and that's a good thing because the NADPH oxidases actually create these radicals to stimulate the processes of healing. One of the things that occurs is these radicals are actually antimicrobial. And so the pathogens suffer from that. But they also help constrict the blood vessels so bleeding stops. In addition, these radicals also contribute for inflammation, and therefore the growth factors and cytokines that are stimulated will bring in the leukocytes that are needed for the repair. And we know that the leukocytes come in a temporal and spatial way, very precisely mandated by the proper healing. So neutrophils first to clean the wound of pathogens, and then pro-inflammatory macrophages to clean the damage the neutrophils did, because they are quite sloppy eaters, and then M2 anti-inflammatory macrophages to set the repair events to make the granulation tissue, and finally the lymphocytes for the remodeling. Well, in the chronic wounds, this whole thing is disturbed. This balance is disturbed because we have excess oxidative stress, and not just ROS, but we also have NRS. So, for example, if you have a lot of superoxide anion that's produced by the noxes, then you have SOD, which is a superoxide dismutase. That dismutate is superoxide anions to hydrogen peroxide, and this hydrogen peroxide is then broken down into water and oxygen by catalase and GPX. So this in normal process, this occurs very smoothly, and the radicals have time to do whatever they do, but then they disappear. Well, if you have excess of it, the dismutase continues to dismutate to hydrogen peroxide, and if the antioxidant enzymes are low, then that hydrogen peroxide in presence of iron can actually give rise to hydroxyl ions, which are radicals, which are very, very damaging to DNA, to proteins. They modify the proteins so they are not functional anymore, and they peroxidate the lipids. Lipids are critical for the membrane, right? Function. Similarly, if SOD doesn't take care of all the superoxide anion, then in the presence of nitric oxide, peroxynitrate ion is formed and is equally damaging to the molecule. So now you realize there's an awful lot of tissue damage, and that is the ideal situation for the bacteria who are present in the skin of us, right? To then install themselves. And those who can survive the oxidative stress by turning on mechanisms that are detoxifying for that, they will stay and install themselves as biofilm forming, while the other ones that are in the microbiome of the skin and keep everything in balance do not do that. So that's why I thought, and then there's a lot of cell death in the wound. When they become chronic, and that's a great deal. We found out through these studies that there's a lot of problems with energy production, especially ATP production. Therefore, the cell doesn't function right. So that's one of the reasons why I began to think to develop the model. How can I do this? And I thought, how about if I take just C57 black and raise the oxygen stress right after wounding? Can I make these wounds become chronic? Well, the animal is normal in physiology, so they got rid of the oxidative stress very quickly. So we thought maybe if we had bacteria, then we might be able to keep this going because the bacteria install itself and forms biofilm. Well, it didn't happen that way. We had in order to keep the wound going in an impaired fashion, we had to treat every week with antioxidants and bacteria, and sometimes the mice just got rid of it. Okay? So just to go retrospectively, I thought, people that are normal don't form chronic wounds. So you have to have some kind of comorbidity, and I chose to use the diabetic model, diabetic obesity, DBDB. So we actually were, I would say, lucky, or maybe we're prepared to be lucky, that once we did the wound and increased the oxidative stress, the wounds became chronic 100% of the time. So that was the ideal model for me, because then I could just go on and do my experiments. And this is what really happens in humans, right? You need to have, there's two critical parameters here. The mice have to be, they have to have the comorbidity, be diabetic and obese, and they have also to have a normal skin microbiome. We can't do this with animals in the clean vivarium. We just can't. So those two parameters were critical, and that's what happens in humans. And I was very lucky to be able to get that going.
Too many people use the diabetic mouse model, but their wounds aren't chronic and they heal relatively fast. They're slower, but they're not chronic, to say the least.
That's right. And they use mice that are 2 months old, and they use mice that come from Jack's laboratory in a clean environment. So the environment is not conducive to that. And that's one thing I was very alert to. We have to make it so it mimics the humans.
There were some great talks at IWH talking about the dynamic interplay of even just hair follicle formation and how you need to have a dirty environment to have that.
Yes.
Be able to produce hair follicles.
Yes.
Really story. Why don't we dig a little bit deeper into the paper? Can you walk us through the methodology utilized in the study and the advantages of nanostring encounter arrays versus RNA-seq?
Sure. So for these studies, we examined the metabolic signaling pathways using both nanostring encounter and WGCMA during the first 48 hours of development of wound chronicity. That is to summarize the whole method. And as for the differences between RNA-seq and nanostring, firstly, what comes across is RNA-seq. For RNA-seq, we need to convert RNA to cDNA, which sometimes introduces amplification bias. Now, RNA-seq is a very powerful tool that generates a massive amount of transcriptomic data. But in nanostring, what happens is because the hybridization probes in the customized array directly bind to the RNA, we can sidestep the amplification bias. Also, the quality and quantity of RNA required for an RNA-seq experiment is about 1 to 2 microgram of RNA with a high quality and a RIN number of greater than 8. For nanostring, we need about 100 nanograms, and the RIN number can be between 2.4 and 8. Another reason is the turnaround time. For RNA-seq, we need a library formation or a library preparation, which is a time-intensive step. For nanostring, the total turnaround time is around 24 to 48 hours. It has also been found that nanostring is sensitive towards low-expressing genes, and therefore overall it can be said that nanostring is cost and time effective and is a specialized tool that can be used for investigating the transcriptomic landscape either as a standalone tool or as a prelogue or an epilogue for a larger RNA-seq experiment.
No, that's great. So better sensitivity, no bias, and fast. It's just wonderful. Can we talk a little bit about the weighted gene correlation analysis of the genes and the process by which the genes are grouped into modules?
Yes. So the weighted gene correlation network analysis, or WGCMA, it's a network modeling approach, and it calculates the correlation between individual genes. This correlation results in the formation of a topological overlap matrix, which can then be clustered hierarchically to obtain closely correlated genes or modules, gene clusters or modules. These modules can then be correlated to individual traits like the experimental conditions, etc., which act as a solution for multiple testing and also as a tangible network of how experimental conditions affect the genes in the module. For the input, we use a data set of the normalized counts of all of the 748 genes that were analyzed using NanoString. And along with that input, to ensure scale-free topology, we used an adjacency matrix was created using a soft threshold value of 10. The correlation method we used was Pearson correlation method, and genes which were highly correlated to each other went into the same module. So it also served as a measure of co-occurrence and co-expression.
Great. This question is actually from Kyle, who's not here today. Kyle does a lot of wound imaging. He actually can do kind of 3D imaging of looking at the kind of the structure of the wound bed without any stains, and it's using algorithms to kind of image. It's just amazing what he does. And he was very delighted to see the focus of the panel of genes associated with metabolic pathways. And what led you to focus on metabolism? To me, it's obvious, but Manuela, why don't you give it a go?
Well, you know, so early on, when you have injury, of course, that situation is the tissue is really injured. So it's very difficult for the cells not to be functioning properly and expressing all kinds of genes. So I reasoned therefore that the metabolic pathways that were going on as injury occurred would be the disturbed pathways. And we wanted to look at things like catabolic, anabolic pathways, stress linked pathways, the actual metabolism of the cell. And the best way to go about that was to take advantage of this new technology of the NanoString, which has this panel that had metabolic pathways. And what we were hoping is that the disturbances we would see in the genes or in the molecules would lead us to figure out what pathways were really disturbed, so we could begin to put in context what is really happening between what's happening normally and what's happening in the chronic wounds. So that's what led me to think about that. And maybe you could say, oh, well, you were lucky, and maybe I was, but I think I was prepared to be lucky. But I also want to go back to one thing that I failed to say to you that I think is actually important for people who would like to use this model, is how does this model compare with human chronic wounds? And we know that human chronic wounds have high oxidative stress, have chronic inflammation, lack of epithelialization. They have poor epidermal interactions, therefore the granulation tissue doesn't form well. Vascularization is one of the things that's really poor in chronic wounds, and then the biofilm. In fact, when I was at Jeff Gertner's, I asked him, what is the most important thing that you see in chronic wounds that you would like to fix? He said, angiogenesis, vascularization. So this is very important. And if you go to my model, actually, and look at the papers published in 13 and 14, you'll see all of these characteristics are there. And the ones related to biofilm have been published more recently in 1920. By my student, Jane Kim, she's the first author, and by me. So I wanted to say this, but you said, you asked me what was happening in the first 48 hours. So we induce oxidative stress. So oxidative stress is high, right? And then what we observe, and we observe this in the bioinformatic studies, and also experimentally, that NRF2, is an inducible transcription factor that is very, very important to stimulate gene expression of antioxidant proteins and enzymes. And so it's downregulated. And if it's downregulated, obviously those enzymes are not going to be present in amounts sufficient to get rid of all that oxidative stress. In addition, the neutrophils that come in, we have shown by some of the bioinformatic analysis that they don't work right. They are not taking care of the pathogens in the proper way, but they cause the damage the same way. So macrophages are coming, lymphocytes are coming, and that's what's happening in the very first 48 hours. A lot of things are there. But in addition, a very important aspect that we also found from the bioinformatics and Proma has confirmed experimentally, although it's not published yet, is that ATP production is very much reduced. Well, if the cells are not producing ATP, means they are not functioning very well. And that's why we see so much apoptosis and so much cell death. And therefore, what this leads to in many regards is to a paralysis of healing, right? In addition to all the other things that are going on that keep the chronic inflammation going. A key issue that we found that's very interesting is that although IL-6 is overexpressed earlier to stimulate inflammation, when it's supposed to help macrophages change from M1 to M2, it's downregulated. So the M2 never really formed. Another very critical thing we find very early on is to do with hypoxia, in which if 1-alpha is not expressed like in chronic wounds, it goes in normal wounds, it goes up. Well, it doesn't. Therefore, the TGF-beta, the VEGF, the PDGF are not produced at the levels that they need, especially for angiogenesis and granulation tissue formation. Instead, what we see is if3-alpha. And if3-alpha is not good at turning on any of these things, but it binds to the hypoxia responsive element that if 1-alpha does. So it competes with it because it's higher earlier on, and if 1-alpha is low, so it competes for the binding, but these growth factors are not turned on and healing doesn't progress. Another very important thing we found out is the high levels of a thrombospondin 1. This is a very good inhibitor of angiogenesis, and so vascularization is really impaired. And finally, two final things. One is a lot of enzymes that degrade ECM are elevated, not just MMPs, but also catepsins and catecholamines. As you know, Ruf Ezerov has shown several times that catecholamines are not good for revitalization. We find in particular that epinephrine is elevated. So revitalization is not going to occur. So the perturbation of these processes combined with a lack of ATP puts the wound in a state of paralysis, essentially.
I love how all this nanostring data it correlates nicely to so many other people's work, like Mariana Tomnik's work on looking at senescence of fibroblasts.
Yes.
Yes. Showing the reduction of ATP, and then you see people working with luminescent devices that stimulate mitochondria to divide and then produce a burst of ATP, then allows the wound to move ahead.
Exactly.
And so all these technologies and all the science, but the problem with other people's work, including mine, is when I do qPCR, I focus in on my favorite 28 genes.
Yes.
And maybe missing the whole story and the bigger picture of what really is going on in a chronic wound.
That's why I hired Proma, because when I saw what she did and she applied for my postdoc, I said, oh, this is the person that I need, because it's not just finding up and down and co-expressed, and it's actually put it in the context of the biology. And that's, I think, the combination of the two of us is what's so good and so powerful.
But it was so funny when I read this paper the first time. I was so astounded how rich it was in data and just the whole idea around the whole systematic approach of looking at the biology of a chronic wound, that I went back and read your paper 3 times, and usually with one read I'm done, and I was like, I should go back and read that paper again. But I just thought it was just a wonderful paper.
It took us 2 years, Mitch, 2 years to put together. I say Proma doesn't have children yet, but I have 3. I had 2 babies. That's what I had 2 babies. This is the product of 2 of my babies. It was so hard to get this paper all together.
Imagine.
Yeah.
I can just imagine. I think we talked a lot about the cells, and we talked about the key changes involved already. What's next? What do you think's next on the horizon to study this pathway? And Proma, before you jump in as well, I want you to know we have a great biometrics program at Alera Health, and if you ever want a job, we would like to hire you.
Thank you so much.
Well, I told Proma, I need you to go on this program because that's how you advertise yourself out there. But she's not ready to go. She's not ready to go.
Whatever you say, Manuela, I would like to hire.
Okay.
Thank you so much for the offer, Dr. Sanders.
Yeah.
Okay. So, in terms of basic research, we have sort of mapped the way of what is important for initiation, but we don't want to stop there, because our knowledge could be very useful when you debride wounds and you create a new tissue, and you could use this to guide your non-return of chronicity. But how about if you go to a doctor and you don't have that possibility? How do you treat a wound that you can't debride? So that's why we're trying to do similar studies. We are right now in the middle of it, to try to understand how it goes from disinitiation to progression to chronicity. And one thing I can tell you already, I told you that by 48 hours, the wound seems to be in paralysis. And it is indeed because by 5 days, we don't have hardly any gene expression that is significant. It's not until 10 days that that gene expression begins to come back. And amazingly enough, has very little to do with wound healing and a lot more to do with cancer. So this is also a very important finding, because Mariana has shown some chronic wounds, some cancer genes being expressed in chronic wounds. We are hoping to put this in soon, but as the other paper is taking time. This is part of what I want to do in the basic science. Also, the other part in the basic science is the microbiome, because we have shown clearly that oxidative stress is important, but the bacteria are very important. Neither one is sufficient. Together, they can create chronicity. So we're working intensely and actually putting together a proposal to study the microbiome, how the bacteria come together and interact to form this complex biofilm, because it's not one bacteria in the biofilm, it's several, right? So we're trying to see how we're going to approach that, and how we use small molecules and antibiotics to try to destroy it. Because it has been my belief since 2008, we had a panel when Paul and I organized the program for the meeting. We had a panel of people, prominent scientists, asking, and we were asking, what do you think is the future for treatment of chronic wounds? And I remember that one of the things that came out is that small molecules were very important. And I believe that's the case because they are cheap, they are off the shelf. And if you combine it with antibiotics, it shows you how potent they can be. Actually, Mitch, if you want to read another paper that I think is fascinating, is by Kathy Lee, by me and Kathy Lee, another one of my postdocs. She developed chronic wounds biofilm system in vitro, and she actually studied how NAC dismantles biofilm, and it's really, really interesting, at least for me. I'm planning to use that.
We actually 3D print biofilms now. We print staph, pseudomonas, and strep pyogenes, and we have these beautiful biofilms that we can form in vitro, and then we put them in the animal model as well. I'd love to collaborate sometime on some of these things. We have, I can't talk about the project, but we have a funded project for small molecules for chronic wound infections. But
very interesting.
It's a very, very small world.
Let me reach back to you. Let me reach to you, because I'm just a basic scientist. I don't have the experience you have. I have other kinds of experience, but this, I think, is going to be a fundamental change for us if we can do that.
The challenge in the wound care industry, especially for these kind of things, is if you have a small molecule and it's acting on the body or acting on the biofilm, then it's a drug, not a device. And so then it's a $10 or a $60 million drug program, not a $1 million device. And so it's much more challenging. And that, all the wound healing studies, the drug studies done 20 years ago, all the failed drug trials.
Yes, I do.
hurt people like entrepreneurs like me because, people don't, we're just now getting a handle on chronic wound healing and understanding why those trials failed. A lot of it was because the trials were designed wrongly in the past. We didn't have a run-in phase, just basic things like this. But now the other thing that we faced in wound care, which was a real challenge with the FDA, is the only endpoint they recognized was healing.
Yes.
So you had a debridement drug or you had something that killed a biofilm, it didn't matter. The FDA wanted you to correlate that to healing, which is
a closed wound, if I remember correctly, a closed wound. But what they don't realize, you can have a perfectly closed wound and underneath is a disaster, so it's going to reopen. So
no, I totally agree.
But in terms of what I'm thinking for applied, I've proposed that in the paper, a bunch of ways of treating. I'm thinking of doing a proof of concept study in humans, but in America is not possible. So I'm actually thinking about going back to Portugal for a sabbatical to try to see if I can work with. I have some contacts there that are very willing to work with these non-invasive. None of the things I'm proposing are invasive. So that we could, in fact, be able to treat wounds and see if we're successful. That's where I'm trying to go.
I'm delighted to be involved and see if I can help with some funding. But anyway, I have to run to another group meeting that I'm late for. I'll be late for in 1 minute. But it's a real pleasure having you on the Fireside Chat. I just love this paper, and I think the work that you do, Manuela, is outstanding. And Proma, you've got a tremendously bright career ahead of you, and please apply to Alere Health whenever she will let you. Okay?
Thank you so much, Dr. Sanders. It's very nice to meet you.
Not now.
No, not now. Take care of it.
We have work to do. But she'll be. But Mitch, she'll be much better prepared when she applies.
Okay?